This video, we're going to talk about inorganic compounds, and in the next section in chapter 2, we'll talk about the organic compounds, the macromolecules. So the metabolites are going to be all the things that your body can synthesize, all the things that they can break down inside of your body. As far as nutrients, we are going to have organic and inorganic nutrients, just like we were talking about at the beginning of the section. What is titled is inorganic. What makes something organic is what it's made out of. What's its backbone? And traditionally, with organic materials, they have a carbon backbone. They are all going to have carbon as their backbone. Carbon will be in there, and then they are going to be covalently bonded to a hydrogen somewhere in there. So carbon is traditionally step one to check off the box for an organic compound, and then it needs to be bound with a hydrogen somewhere in there to make it an organic compound, because CO2 has carbon in there, right? But it's an organic compound. It wouldn't count. It is an inorganic compound. Sorry, CO2 is inorganic compound. It's not going to count as an organic compound. So organic has carbon as a backbone, and it's going to be covalently bound to hydrogen. And the macromolecules that we're going to talk about in the next video: carbs, lipids, proteins, fats. Carbs, lipids, proteins, nucleic acids are all going to be macromolecules that are organic, that our body's going to make, take in with our diet, and we're going to synthesize. So organic stuff, carbon. Inorganic stuff typically lacks carbon as its primary structural ingredients. We're going to talk about acids and bases and water, which is super duper important for all of us here. Those are the ones that we're going to talk about in this section. So water has lots of cool properties. It can be used as a lubricant. We mentioned that the heart is beating all the time, and that every time it beats, right, it's moving, and we didn't want it to rub up against other structures every time it's beating. So it's surrounded by this bag, the pericardium, and inside the pericardium is fluid. So it's going to be lubricated by that water in there, so it can act as a lubricant or also as a cushion. We all have joints, and part of those joints, some of the ones with more movement, are synovial joints. And so there is a synovial cavity, and inside of there is synovial fluid that helps cushion the joint that you are going to have there. So water is a lubricant. It can act as kind of like an airbag, but with water inside as a cushion. Another cool thing about water is that it is a heat sink. If you have ever, what does a heat sink mean? It means that it is going to take and need significant quantities of energy for it to change temperature. It is a heat sink, so it is going to absorb and dissipate heat slowly. So maybe not this year, because spring break was a little up in the air this year, but if you were fortunate enough to go visit the beach on spring break for college, you know, the air is warm and it's sunshiny, but you get in that water and that stuff's cold, and it's cold because it's a giant body of water. And just because it's warm outside does not mean that that giant body of water has heated up to that level yet. Water is pretty steady, and it takes a lot of energy to change that. So it's a heat sink. It has a high heat capacity, the ability to absorb and retain heat. Anybody who's tried to make spaghetti or ramen, and you're staring at that pot and wishing that that stuff would boil, you know it takes a long time and a lot of heat and energy to go into it to finally change the temperature of the water and to get it to boil. It has a high heat capacity. It is also one of the ones that is present in our environment in a range of temperatures that are compatible for life. We can tolerate, not for very long times, freezing temperatures where water is a solid. Water is a gas. Water is a gas around us all, water vapor, and preferably temperatures where water is a liquid. So it's in all three states of matter. Water is in temperatures that are compatible for life. When we break the bonds and it becomes water vapor, it takes a lot of energy away from that. I mentioned that when we get hot, right, one of the things that we do is we sweat. So when you break those bonds, as the water is breaking and going from a liquid to a gas and steaming off of your body, it cools you down. It takes that heat with it. So it is a heat sink. It's got a high heat capacity. It is also an excellent solvent. Water is an excellent solvent. I don't know why it continues to stop doing its job with me mirroring the screen, but hopefully I can get it back on there. We can get it back up. There it goes. All right. So it is an excellent solvent. What do I mean when I say solvent? I'm going to need y'all to know the difference between solvent and solute. What does that mean? So water is an excellent solvent. It is easy for us to dissolve things into water. So I love tea. I had mentioned before that I've taught all my kids to make their own sweet tea. And so you take water and what do? You put tea bags in it and the tea will diffuse through the water. Water is the solvent and the tea bag is the solute. I also like sweet tea because I was raised in Alabama and we drink sweet tea. So you got to put the sugar in. And I was mentioning that you got to put the sugar in when the tea is nice and hot because temperature is going to help that sugar dissolve in there. The sugar would be a solute. Solutes are the small things that get dissolved into a solvent. The solvent isn't always necessarily water. It could be alcohol, but that is your solvent. Solvent, water is an excellent solvent. Why is it an excellent solvent? Because of the structure of water itself. We mentioned previously about bonds and how things were bonding in covalent bonds and how water, H2O, is a covalent bound molecule. But we talked about how hydrogen and oxygen are two different sizes. Hydrogen only has the one proton, the one electron in it. So it's small versus oxygen that has a lot more protons and electrons in it. So oxygen's bigger. And so since it's bigger, it hogs the electron on its side more often than the hydrogen gets it. It doesn't share equally like a covalent bond should. It shares unequally. It does share, just not nicely. It's kind of a jerk about it. So it keeps the electrons on its side, therefore it becomes a polar molecule. So because water is polar, this side that hogs the electron more, the oxygen side, has a slightly negative charge. And the hydrogen side over here is going to have a slightly positive charge to it. So water is polar. On the poles, on the different ends of the molecule, one is negative and one is positive. So because it has that chemical aspect of it, it bonds well with lots of things. It is a great solvent. So we can see here we have salt. Salt is sodium and chloride. And the chloride is negative. It's Cl negative. It has pulled an electron. Therefore, it is now a charged ion. And it is negatively charged. So cation or anion. Anion, negatively charged ion. So chloride is negatively charged. It's got an extra electron. That negatively charged chloride is attracted to all of these positively, slightly positive, because they're not fully positive, they're slightly positively charged hydrogens. So we got hydrogens coating the outside of the chloride and it forms something called a hydration sphere. On the other side, we have busted apart salt. Salt is sodium and chloride. The chloride is covered with the hydrogen sides of the water. And the sodium side, sodium lost its ion. It gave it to the chloride, and therefore it didn't lose its ion. It lost its electron. So since it lost its electron and gave it to the chloride, it has more positive protons than electrons. So we are positively charged because our electron number is less than the positive protons. We have a positive charge, so it is cations, remember, with a positively charged guys. So sodium has a positive charge over here. And since it has a positive charge, it's going to also attract water. It's just going to flip to the other side of water, and it's going to be attracted to our slightly negatively charged oxygens over here on the edges. So water is an excellent solvent because it has those polar properties, because it can attract both positive and negatively charged ions. It's an excellent, excellent solvent. And our body fluids are going to have lots of water. We have lots of water. We're made up of a lot of water. Later on, when we get into AP2 and we go into the fluids chapter, we'll talk about how guys versus girls and exactly how much water we are made out of percentage-wise. It is not 99%, by the way. It's closer to 50-ish and 60-ish percent, depending on guys or girls. But regardless, there's a lot of water. We got a lot of water inside, and it is an excellent solvent. Don't get solvent and solute confused. Water is the solvent, the things that stuff goes into. Solutes are going to be the little guys that get put into the water or the alcohol, whatever the solvent is. Solutes would be the tea bags. Solutes would be the sugar crystals. Solutes would be the Kool-Aid packet that my kids like to rip apart and dump into their bottle of water, or the Propel packet. Those are solutes. Don't get them mixed up with solvents. Moving on. It is very reactive. Hydrophobic and hydrophilic. If we have learned our terms that we needed to know from chapter 1, right? We know hydro means water, and phily or phobia. Phobia, I hope we all know before we ever got to this class, if you have a phobia of something, you're afraid of something, right? I don't like spiders. It's irrational. I know they're small, but like I just don't like them. I'm a wildlife biologist and I don't like spiders. I admit it. I don't like them. They give me the willies. I respect them. I don't kill them all, but I don't like them because I can't get a feel for what they're doing or if they're upset or happy. I don't know. Can't get a read on spiders, so I don't like them. I have arachnophobia, because I don't like them. Snakes, I'm okay with. Bears, I'm okay with. Spiders, ugh. Hydrophilic. Philly, the city of love. Philadelphia. Philia, you love it. So hydrophilic are going to be our polar things because we just saw sodium chloride is charged and it loves water and it's going to bind with water. If you are hydrophobic, those will be non-polar things. What's hydrophobic? What doesn't go with water ever? They never mix. They're always separate. Oil, your lipids. This, if you eat salad and you have salad dressing, you can see in the dressing container that the oil and the water, they're never going to mix because oils are hydrophobic. They don't like water. While things that are hydrophilic, like the vinegar, those guys do like water. So know the difference between hydrophobic and hydrophilic. We'll talk about that for the rest of the semester too. What about concentrations? We will talk about concentrations. Remember we talked about homeostasis, that you have to have something that's in its happy place, and there's going to be a range of things. So blood glucose is, here we've got a range. We have a measurement of 100 milligrams per deciliter is a measurement of blood glucose. And we can, in this range, things are okay. This is your range. If we were talking about glucose, maybe we're talking about your pH for your blood. That's 7.35 to 7.45. There is going to be a range in here in concentrations of different things. Maybe we're looking at glucose. Maybe we're looking at sodium. Maybe we need to know what the potassium levels are in somebody. And when you run a blood panel, you're going to see all those numbers. I'll have one of those for you in AP2 when you get there. But you can see all those numbers, and it actually gives you a handy dandy range. So you can just look and see what your patient's stuff is. Does it fall within the range, or is it above or below the range? And you'll automatically know just by looking at where they are and what the range is supposed to be. We need to know what those are. Another measurement can be molarity. That is something that I would have my majors bio people calculate. I am so not going to have y'all calculate molarity, but that's going to be the atomic weight of a substance dissolved into 1 liter of water. That's how you measure it. If you had chemistry already, you know about molarity and Avogadro's number and all that good stuff. With molarity, just basics of it. Here we've got glucose, C6H12O6, and we said that those little subscripts right there under the 6 for the C means that I have 6 carbons. Well, if I know that the atomic mass of carbon is going to be, what is carbon's number? I think it's 12.0111. 12. If I have carbon and its atomic weight is 12, then I have 6 of them. So then I would do 6 times 12, and that would give me, what, 72? 72, yeah, in my head. 72. And then I would do the same for hydrogen. Hydrogen's atomic weight is 1, and then there's 12 of them, so I do 72 plus the 12 hydrogens, so now I'm up to 84. And then I do the oxygens, and I cannot remember what oxygen's atomic mass is off the top of my head. I'd have to check the periodic table and see, but then I would do 6 times whatever that atomic mass is. I would add it all up together, and that would tell me how many grams of this substance would be dissolved into 1 liter of water, 1 liter of solution. So if I was making a 1 mole solution of sugar water, glucose, like I had a table of glucose, a bowl of glucose here. I got a bowl of sugar, and I'm just going to scoop it up. I'm going to put it into a liter of water because I'm going to make sugar water. I need to know how many grams of stuff I need to scoop up. So I need to weigh it out. How many grams do I need? I calculate up the molarity, and I'm able to calculate that. And I've done it before, and I've got it in my notes, and I could tell you, what is it? 180. I would need 180 grams of glucose that I would scoop out, put onto a scale. I would take that and I would put that into roughly a liter of water, and that would give me 1 mole solution of glucose water, of sugar water. That's molarity in a nutshell. I am so not going to make y'all do that. That's more of a chemistry majors bio kind of event there for you. Different types of solutions. There's something called a colloid, and that is when the solution is, I'm hoping this is still recording, that the solution, I think we've lost it. It's not skipping ahead. All right, let me check again. Okay, so types of solutions. The colloid, with those guys, those are when the proteins get suspended, and it's almost like Jello. Pretty much it's like Jello. Those proteins are suspended throughout, and so it's a Jello-like substance. It is Jello, pudding. I don't know how else to describe that, but you know what I'm talking about when I'm talking about Jello. It's like a semi-solid kind of substance. That's a colloid, and you do have that kind of type of substance that happens in your thyroid. Thyroid is going to make a colloid-like solution that gets in the thyroid gland when we start to produce T3 and T4 when we get iodine in there. Iodine is one of your trace minerals that we mentioned before that you need in your diet. And that helps you to produce T3 and T4 that's coming from your thyroid gland. And those help with your metabolism. So we got thyroglobulin, is that showing up? And iodine, and that goes on to make T3 and T4. And these are the chemicals that help speed up your metabolism. The more T3 and T4 you have, your metabolism will go a little slower. If you are hypothyroid, your thyroid's not producing enough T3 and T4, your metabolism's super slow. So colloid is a jelly-like substance. A suspension, on the other hand, is not a semi-solid. It's not spread all throughout. On a suspension, things will settle out of solution. So a good example of a suspension is blood. If anybody is in the healthcare field and you see containers of blood, you know that the stuff will settle out, that you'll have the lighter liquidy stuff on the top, and then the heavier stuff will settle down to the bottom. And you can exacerbate this if you stick it in the centrifuge. So the centrifuge, you'll take your blood, I can't remember the name of the capsules that you put them in, and then you stick them in, you make sure you put the top on top of the capsule, and you turn on the centrifuge, and it's basically just a giant spinny. If anybody's been to the circus or the fair or Six Flags, right, and you ride those rides that they spin, so everything that's spins out to the side like that, and the heaviest stuff goes on the outside. So if I'm riding the ride with my kid at Six Flags who wanted me to go on the one that goes like 200 feet in the air and you're suspended in that swing on a chain, so I'm the heavier one out of the two of us. And so I go out to the sides the furthest, and my lighter child is closer to the inside of the event. So heaviest stuff spins out to the side, and the lighter things go on the inside. So that's the way it works here. So we've got our red blood cells when we spin down the blood. All of this is red blood cells. And then this little white buffy coat right here, these are the small amount of white blood cells that you're going to see in everybody's blood. How is your white blood cell count? And then the rest of this stuff is liquidy plasma that's filled with proteins and other stuff in it. But the bigger things, the red blood cells and your white blood cells, those things settle out and go to the bottom. That's what a suspension is. Things can drop out of suspension and settle out. So there are colloids and there are suspensions. Water is reactive, most definitely. We mentioned it is polar and it likes to stick on to charged ends of things. We mentioned how we could synthesize and create something new, or we could decomp and bust something down. We mentioned dehydration synthesis. That was this one because we pulled out H2O. We pulled out the water, we dehydrated it, and when we did so, we synthesized this new molecule. And if this was a sugar, this is a monosaccharide, and this is a monosaccharide, and now we have made a disaccharide over here. On the other end, we used water to bust stuff up. So here's our disaccharide, and we added water to this monomer molecule, this disaccharide, and now it is split apart. And we've gone from a bonded molecule that we've busted up using water. And we remember from chapter 1, lysis means to rupture or burst. So this is called hydrolysis. Hydrolysis happening here in this reaction. All right, salts. Hopefully this picture is showing up for you. These guys, salts are going to be electrolytes, and your body needs electrolytes. You definitely need salts. When electrolytes are broken up, if they are not releasing hydrogen or hydroxide, no, other than not this guy and not this guy, when they dissociate, when they break apart in solution, because we put salt into water. Remember, water was polar, so it's going to pull these charged things apart. When they break apart, if they are not releasing hydrogen and they're not releasing hydroxide, they're a salt. They're a type of salt. So we have sodium chloride that we already mentioned. That's a type of salt there for us. There is going to be other things out there that it's not always sodium chloride, but that is one type of salt that we have mentioned before. All right, so those electrolytes that I said we all need, they're going to be soluble. They'll dissolve in water. Inorganic, meaning that there's no carbon backbone on these guys, and the ions will conduct an electrical current. If we put some sort of, if this is the solution and we put a positive end over here and we put a rod in here with a negative charge on this side, these guys would split apart in solution. So when they split apart, they can conduct electrical currents. Remember, they are going to be ions, and who's a positive charged ion, who's a negatively charged ion. Cations were the positives, anions were the negative. So sodium is a positive, while chloride ends up with that negative because it's got the extra electron sodium gave away to chloride. There are other non-electrolytes that are essential for your body. Of course, we need sugar, we need glucose. We are also going to be producing urea, and we're going to have proteins and lipids and creatine in here too, as non-electrolytes that we're going to see in our body. pH scale is definitely something I want you to know. We'll go more in depth on pH scale in AP2 when we go into the fluids chapter, but for sure, let's talk about basics of the pH scale. So neutral is 7. Pure water is supposed to be at 7. If you have filtered your water through a reverse osmosis filter, you have pH of 7 water. So 7 is perfectly in the middle and it's neutral. Anything above this, anything higher than 7 is going to be called a base. It's also called alkaline. So let's write that down. Alkaline base. I don't like the name base. It works and I know what it means, but it's kind of tricky because when I think of the word base, I think of the lowest number possible, the bottom of the barrel. And that's not the case for the word base. In this instance, the word base is going to be the upper level of the pHs. Base is going to be anything greater than 7, all the way up to 14. The other end of the spectrum, on the lower end of the spectrum, these are going to be acids. All of the acids are going to be less than 7 on the pH scale. The pH scale is logarithmic, meaning it goes down by powers of 10. So it is going down by a power of 10 when you go from 7 down to 6, and then from 6 to 5. So we're going down every time, logarithmic, as we get down to lower pHs. Hydrochloric acid, a very low pH. Here it says it has a pH of 0. I've been told it has a pH of 1, so it varies, but it is super duper low, and that is what is in your stomach. Your stomach produces hydrochloric acid to dissolve your food. It's also helpful as your immune system because it helps dissolve some of the bad guys that we don't want getting sick. So our hydrochloric acid helps us in our stomach. As it leaves the stomach, we have to buffer that because we don't want to burn up the rest of our intestines on the way out of our body with the hydrochloric acid. So we're going to neutralize that. We need to get it closer to neutral. And which number was that? 7. 7's neutral. Above it is a base, and below it is an acid. Above it, alkaline. Below it, acid. They are done by powers of 10. Over on the alkaline scale, at the top, we have examples of this. Bleach. Anyone who's doing your own laundry, right? If you are doing the whites and you need to add bleach to it, and you get your bleach on your fingers or soap, any of that stuff, those are alkaline and they're kind of slippery feeling. Those are highly alkaline. They're going to have high pH numbers, 14, 13, 12. Those are highly alkaline. Your body's blood has a pH of 7.4. It's anywhere from 7.35 to 7.45. So if you average it together, 7.4. There's a question on the practice quizzes from WiseWire asking you about a patient whose blood is 7.48. 7.48. So is it higher or lower than 7, which is neutral? If it's higher, it's a base, right? And if it's lower, then it'd be an acid. So what's 7.48? I'm going to let you take that question on WiseWire, and you can make the judgment call on what you think it is. But anything above 7, alkaline. Anything below 7, acid. When you are considered an acid, why does it even have that name? Everything on the acid end of the scale, when it breaks up into solution and it busts apart, these guys are going to be proton donors. So when I mean proton donors, when they break apart, they release hydrogen. And oftentimes hydrogen just has, it's H+, it's lost its electron. So it's literally just a proton, hence the name proton donors. So acids, the stronger the acid is, the more it releases hydrogens in solution. So hydrochloric acid, when it breaks apart into hydrogen and chloride in solution. It's a very strong acid. Proton donors are acids. On the other end of the spectrum are the bases, and they will be proton acceptors. Oftentimes it will be hydroxide. It's not always hydroxide, but oftentimes it is. So OH negative. And what happens if OH minus gets another hydrogen in there? What do you make? We got 2 hydrogens and oxygen. You made water. So on this end, these guys are proton acceptors. They are bases. And on this guy, hydrogen donors, they are acids. Acids. We'll talk more about this in AP2 and fluids. This is the basics of pH scale. It's logarithmic. Where is neutral? What does it mean when I'm telling you it's an acid? What does it mean when I'm telling you it's a base? Who's a proton donor? Who's a proton acceptor? Here is more descriptives about them. Again, acids, they release the hydrogen ions called proton donors, and they are anything less than 7.0. So here's an example of a strong acid, hydrochloric acid. Here's an example of a weak acid. This is carbonic acid. Later on in AP2, we'll talk about how you can buffer things. When I say buffer things, again, it means to get it closer to neutral, because you don't want that super strong acid burning your guts out on the way out of your body, right? You've got to buffer it, get closer to a neutral pH. So we want to buffer things, and one of the ways that we buffer things is we can make a strong acid and turn it into a weaker acid. We can turn something into carbonic acid. Carbonic acid is going to be something that we can break apart, and it will change into a hydrogen or bicarb, or it can change into H2O and CO2. And then we get to the lungs and we exhale out the CO2 and we're just left with water. So carbonic acid is a weak acid, and it's helpful in our buffering system later when we talk about this in AP2. Bases, again, alkaline, they may be releasing hydroxide, but they are going to be proton acceptors. They want, gimme, 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 the protons. And they are going to be greater than 7.0. Sodium hydroxide is an example of a base. And when sodium hydroxide breaks apart in solution, we have sodium, and then we have hydroxide. So this is most definitely a proton acceptor. This guy wants the proton, this guy wants the hydrogen, so then it can make H2O. So these are bases. What pH range is base? Above 7. Everything above 7 up to 14. Acids versus bases, when we put them in solution in the water, and look, we got hydrogens, acid or base. Here we have hydroxide. These are going to be base or alkaline over here. Buffers gets you closer to neutral. There are lots of different types of buffers. There are protein buffers and bicarb buffers. They are used inside the cell and outside the cell, and some go in between both of them. But what we want is to take something and get it closer to pH, whether that is a strong acid or maybe it's a strong base. Either way, we want to buffer it closer to 7. That's our happy place, 7.35, 7.45, right? So we want to buffer it. So we'll talk more about this later in the fluids chapter in AP2, but we could use sodium bicarb, or we could use our carbonic acid like I was telling you about, and change this up to where these substances are not quite as strong when we add those buffers in. All right, so we will go through organic compounds in the next video.